it's a great honor to be here, and 30 minutes, probably I tried briefly to give a report of my team research result and on the pathogen research. Well, before I start, I was asked to, give a short speech after our Vice President Hong Wei-Wang. That he didn't appear, but I said I have to give a talk, so I shouldn't say anything. But after think, I thought I should say something about And in the spring this year, I was in Oxford. I was in UK, and where I delivered talk. The title is "A Tale of Collaboration Between Sino-British Scientists Last 60 Years." Well, I think this, as a scientist in the world, we should remember even during 60s last century, the scientists between UK and China, and through Royal Society and Chinese Academy, they has very extensive collaboration, and I show quite a lot pictures and show that important history. So I'm so glad that our American Microbiology Society has a meeting with us in Tsinghua. I pay tribute to you, and we need cooperation. COVID pandemic, the lesson told us already, we need collaboration. Okay, back to my talk. And over the last 3 decades, I can see more than 4 decades, and since I was in Oxford, and I've been dedicated in the pathogen research. My work started in flu and last century, late 80s. So we want to answer 3 questions, but perhaps not in my life. And but the first, the entry. This is something is very crucial, and second is transcription and replication. Third is assembly and release. Those work last 3 decades. We through our work, structural biology work, we did understand something. We find something and exciting. Well, this is one of the list of our team Rao labs and the work. You can see the start from influenza virus. So I just give you a glance of that. And I mentioned before, and when I was in Oxford, and with David Stewart and George Branney, and the pioneer of flu person, his postdoc, Greg Winter, and so I started work on the structure of influenza virus nuclear protein. That anyway was, if I stick on that work, then probably I'm not be here today. So I have to change job. So after 3 years postdoc work, I got nothing there from there. So I spend a lot of money and have a big lab record and lots of things. And I can say I learned a lot, and this is a starting point for me into the pathogen research. And then coronavirus. But I should continue to say something like that. The flu. Then when I come back to China, 1996, I continue this work. And until 2008 and 2009, I published 2 Nature papers in the transcription polymerase structure on that. There are 2 Nature papers. So recently, we still work on that. So with this RNA virus and for coronavirus and influenza virus, they are probably flu is has high impact than COVID. But so we used technology and the platform for coronavirus research. We shifted that to the flu. We got exciting result on the RNP. So I hope you will see the result published somewhere. And this work being done by Zhihe Rao and in Tsinghua University. So now I'm under his leadership because he's leader of Tsinghua group. Then coronavirus, since 2003, SARS outbreak, and we start COVID research. So fortunately, we consistent for work until COVID-2 breakout. So we benefit a lot. We've been very fruitful, very productive on this area. Anyway, and I'm not talk too much on this area. So the flavor virus, this is a famous one. So, but we have solved the JEV virus structure. We have been working quite a lot on this flavor virus, and then the piconovirus. I think we in collaboration with David Stewart, and we have been very fruitful in this area, and we solved the hand-foot-mouth disease virus and the EV71, Kasaki 16 and 10, 6, and so on, and especially with the receptor. So we, from this structure, we understand a lot on the virus entry. So this is not main topic for today. So, and one is proud of things, this is hepatitis A. So, just we've been working on that hand-foot-mouth disease virus. Then we just by chance solved the structure of hepatitis A, this which appeared in Nature in 2015. And so we understand quite a lot on this one. And rabies virus, and this project have been worked more than 10 years. So recently we, in collaboration with Hongwei Wang, and we have some exciting result, methodology and biologically. And this we also want the paper showing somewhere CNS, I hope. And anyway, recently we have, just 2 weeks ago, and this paper appeared in Science. And hepatitis B, this is the main, well, I will talk a bit more on that later. And 2019, we published African swamp fever virus structure and in Science. This is probably the largest virus structure being solved in the world. 2018, we also published herpes virus structure in Science. At that time, we thought this is the biggest virus structure at atomic resolution in the world. But only after 1 year, the African swamp fever virus structure solved. This one is about more than 10 times bigger than the herpes virus. Anyway, and African swamp fever virus, the main purpose for this work, we want vaccine. This vaccine work and being doing extremely well. And we hope we can have vaccine food industry and the agriculture industry. Herpes virus, we not only solved herpes virus structure itself, but also solved the terminates, the carrier of the DNA, the packing, the machine. So with these two structures, we have more understanding on that virus assembly. But I have to say, for this influenza virus and the SARS-CoV-2, this RNA virus, for the replication transcription work, so we have some new understanding, new finding on this transcription replication. Of course, this part work now being done by Zihe Rao. And I think 15 years ago, we started working on that bacteria tuberculosis. So everybody knows how important, why I'm being involved in this area. I hope we can, by the targeted research, by the structural biology and the targeted research, we want, in collaboration with other people in the other area, being together, being joint force to discover a new generation anti-TB drug. This is what we want. Well, and so this is main. Before, my team being solved delivered so many viruses, structures, particles, and protein machines. I think my team is probably one of the most active, productive team in structural biology field. Okay, and for the tuberculosis, we also very proud. So we are teamwork. So always said, every road to Roma. So we have team, and make a paradigm not clash to each other, but sometimes we have to cross, collaboration and associate with each other. But so we have the team work on the cell wall synthesis system, host pathogen interactions, and the energy metabolism system, gene regulation system, and secretion and transport systems, and also the cross-scale whole cell imaging. So, of course, with this, we need collaboration with clinical medicine and international corporations. So that means we have 8 small research groups, everybody working on one road. So this is all road to Roma. So after extensive work, so I still very proud. So my team probably is one of most active, most productive team in the world in TB drug target research. So, at the beginning, when I started this work, and I was the president of Tsinghua University, so I have dedicated my 99% my effort in my job. So I only can spend spare time with my research. So then that time, my student, the whole bacteria has so many proteins. So yes, most of them, or some of them easy, some of them difficult. So without monitoring, so the students solve structure like enzyme in a soluble protein. They can publish a small paper and get a degree. So when I stepped down from my position, I become tough. I said, well, we need to do membrane protein, cell wall synthesized protein, no soluble proteins. We need to do protein machines. So otherwise, you cannot complete your PhD thesis. So actually, this worked. So 2018, we published in the energy metabolism, that super complex structure in science. This is only, will take us about 4 years effort and get this structure done. And not only this super complex, we also find one of the major things, what that? SOD. You know, for the energy metabolism, SOD play very important role. But how can they associate with? Actually, this is something, you know, it's the diamond on the crown. So we found a SOD bind on this super complexes. Well, after that, we have a paper published, Cell, Science, Nature. Recently, only a few months ago, we have complex 5 and with drug, beta-quinine, and the mechanism to their interaction, and this paper in Nature. Not only this, so I think the work, systematic work, and one, you know, whole complete work in one area, is very difficult, you know. But we done. We still carry on. But now, my group from the people ask us sometimes, Professor Rao, you're working on TB. Yes. I said, we didn't find you have applied any ground on that. I said, why? So what? So, and then we now, and people knows, and we have wide collaboration with the clinical work and pharmaceutical companies, even Bill Gates condition. Well, I mentioned, I'm so proud, you know, hepatitis A is, well, we just solved it by chance, in collaboration with David Schultz's group. Well, after we say 18 years, actually it's much more than 18 years. But on this project, with this material, we've been seriously, you know, struggling on last 18 years. Of course, we very delighted, and we got this structure solved. So this was a press conference in the September 13, and actually the paper, of course, after paper appeared. Of course, the team, you know, the Tsinghua Institute of Biophysics and Chinese Academy of Science. So the Nankai University, Shanghai Tech, Guangzhou laboratory. Well, this one is tough project. This everybody knows. You can see the you know, morphology, and this is DAM particle, filaments, and surface antigen spheres. For this one, we start with this one, but of course we would like to get into that piece in the category 3 NAP and with cryo-EM. We want to do something more, and this is on the way. Anyway, we thought this should be okay after, you know, and I'm a crystallographer. So we thought the crystallization is the way to the purification. This everybody knows. So, and when I get this this project, and we, first things, I said, try to purify it, try to get it crystallized. So as soon as it can be crystallized, that we have hope. Well, something talked later, okay. So for the hepatitis B virus core antigen, actually I also got crystals when I was in Oxford, but the structure, there's that structure being solved and by Alistair, Stephen, NIH, Richard Crosser in Cambridge. This is end of last century. Then crystal structure important, and they solved it by Andrew Leslie in RMB. And but they are icosahedral. They can crystallized. They can be crystallized. The crystal can diffract to 3.3 angstrom. That's good. So the structure solved, and so this is icosahedral. So icosahedral normally, easier. Well, for the surface antigen, S particle, S protein, this is one we call the hepatitis B surface antigen particle is one of the most famous unsolved structures in structure biology. This and it's a half-century enigma since being discovered. And people spent a lot of effort, like, and Mark Eager and David Stewart and Zhu Ping, they've been worked extremely hard, and they get something, but not quite. But, well, and 2006, we got a sample from Professor Hu from National Institute of Food and Drug Control. We got this sample, and then we try to purify them, So, and at the beginning, I thought I was lucky, and we got crystals. We have a Ph.D. student, and he been working on this and get crystals. We take this to diamond, took that to diamond, and unfortunately, they only diffract to 10 angstrom. But of course, they still assist. So, and it's a bit disappointed, but this egg crystal, then this guy now is the mayor of one of the city, famous city, Hainan province, Zhuhai, he's mayor. But of course, he is also an author, co-first author in this science paper. And so after we got cryo-EM set up, so we start collecting data. So before I thought African swine fever virus, because it's too big, so one imaging probably only can collect one particle. So we collect lots of data for the African swine fever virus. But this one is small. But we have the data collected and accumulated more than data we got from African swine fever virus. You can see that there's morphology. It's very difficult. This sample we got from dissolved crystal. We get a sample, we crystallize, we suppose that is one of the major way to purify it. Then after purified these crystals, we find they are still not good, not quite good. So this is the reason they cannot diffract to, and good resolution. Well, I have to mention last 5 years, I have a genius student called Wang Tao. This is a Tsinghua student. He been working on this project. So, and he developed one of the computation methods to classification and to measure the diameter of particles and whether they run enough. So actually, one day he told me, "Wow, this is not fair." Some like I said, "Well, like rugby, is that right?" Yes, this is this in the high resolution they have very different morphology and arrangement. So sometimes people say, "Well, in this case, you never get the structure." Luckily, after struggling, hard working, the teamwork. So, and we finally solved these two structures. We got two structures with different symmetry. One is the, we call it D2. D2, actually, D2 is just with two-fold symmetry. D4, this is a D4, this is a four-fold symmetry. Yeah, of course, we solved it, and the spike actually is a 2S protein dimer. The S protein structure and dimer. So, and this is one of the, it's not a regular way for structural biology. Oh, time is flashing. And this is why I said I pick up the right person to do right thing. This structure, if you use traditional, normal way to do the structure, you never get it. So anyway, we did that disassembly the particle data and reassembly and superimposed, averaging. So luckily we got a 3.7 angstrom S protein structure. It's a new atomic resolution. You can see the side chain, quite clear. So these people always want to know what else it look like the S protein. This is major surface antigen. But unfortunately, they are still, by modeling all of S protein on the sphere and aligning them, we find that there is a considerable structure heterogeneity among this S proteins on the surface. You can see that they are the same. They are not the same. They are like the body is the same, but their movement that makes things different. Well, this is a dimer. So this is the surface antigen spike. This is spike, they just hugged each other tightly to form a dimer. This has a very extensive interaction with the two dimers. Okay, you can see, even on the dimer is not enough. They have to be oligomerization and form a four type of oligomers. You see, different oligomers. So this is oligomers on the surface of this is a antigen particle. You can see there the oligomers still shows great heterogeneity. You can see that. They are still dancing. So this is makes things difficult and okay. Okay, and jump to here. We got structure. You see, this structure, with disassembly, this structure, for this loss pole, it has two caps, one belt. This is the top of cap. This is, bottom of cap, the former cap. This top, see the cap. This belt. So, two caps, one belt form a sphere. So this sphere you can extend and means two cap, two belt, two cat, four belt, two cat, and belt. They can form into filament bars. You can see that, filament particles. So amazing. This is, Okay, this is a movie. I don't think we have time to, I think it's already quite clear. This is how this structure like. So I jumped this because then, of course, this is the N-terminal pre-S and the HB, the virus envelope may interact with the internal core through the 2,4 axis. So this is the conclusion. So I think the major things we already see it clear. Then we got the structure. This is unsolved, but now we have that. So this is a very important start point for vaccine and drug, treatment vaccine and drug research. Okay, this is being 2006 and Institute of Biophysics, then down to the Nankai University and Tsinghua University, then Institute of Biophysics and Shanghai Tech University, and get result come out. And thank you.
Thank you, Dr. Rao, for that fascinating talk. We have time for one question. Okay, I'll ask you. Why do you think hepatitis B was such a difficult structure to solve?
Well, because they have a different morphology. They are not icosahedral. They don't, even in the same particle, they have different arrangement. So this is one of the problems in the virology area. So icosahedral is okay. Non-icosahedral is difficult. For this one, it's, too small. You cannot do cryo-ET. So, but anyway, we saw.
Beautiful.
Thank you.
All right. Oh, quick, another question.
So, Dr. Rao, very interesting talk. So what decides or determines such kind of, complex shape or different size of the particle for HBV? What kind of subunit, different subunits are the?
Well, I think that the basic compartment in the antigen, they're same. But the arrangement, the assembly, they are different. But they must be their reason. We cannot answer it for the moment. But for this disease, so severe, this we wish this is nature. Okay, this is nature. So we are now getting confident, to attack this sort of problem.
Okay, let's thank Dr. Rao again.